So who's having fun? Yeah, I am too. This is really, really great to be with you all. And so strap on your 5-point harnesses. Get ready for the old Jeff Bland. No, I'm just kind of kidding. We're kind of developing a new model, hopefully slowing down the pace slightly. But I will say there's a lot of news to use in this next section. So I hope your cognition is fully activated, your neurons are oxygenated, and you're ready to rock and roll, because here we go. So I'm entitling this presentation From Metchnikoff to Mainstream. And those of you who have been around in this field for a while know exactly where I'm going. I always like to put a historical context on information because I think it helps to tell us where we are from where we've been and where we're then going to go. So this is kind of a quick historical review of how we got to where we are today with the gut microbiome and where it may be taking us, the fascinating fall and reemergence of this GI-centered medicine that we're witnessing come to life around us the last few years. So let's go back and just revisit Ehrlich Metchnikoff for a second, because I think that his contributions to this field are quite considerable, and some people would actually say that he is the father of naturopathic medicine, because the naturopathic model is it all starts in the gut, as you know. And so Ehrlich Metchnikoff is a fascinating character in history, in medical history. He's a Nobel Prize winner of medicine and physiology in 1908. And he obviously is Russian, but he was a Russian physiologist who later came and worked in France and took over the Pasteur Institute as its director when Louis Pasteur died. And so you have to put it in the context that he was in the epic period of microbiology, the development of the concept of microbiology. And remember, in the late 19th century was when we really started to see the concept of infectious disease being associated with these corpuscles that we call microbes. And the microbiological world started to be more well understood with the advent of the microscope, and then trying to do the taxonomy of these organisms and starting to recognize they had something to do with illness, with infectious disease. And so the story goes that Metchnikoff was at the southern shore of France, the seashore, playing around at low tide in the tide pools, and picked up a starfish and a sea urchin. And he took the sea urchin with its spiny quills and he punctured the starfish. And then he had his monocle, his eyeglass, and he watched at this point of the intersection of the spine, and he saw these corpuscles coming to the point of the injury on the starfish. And of course, being the start of bacteriology, he immediately assumed these were bacteria somehow coming to the point of injury. But then in further study, he found out, no, they weren't bacteria. These were endogenous things that looked like bacteria that he called macrophages, right? They were organisms that would eat debris, macrophages, which then became the innate immune system. So he discovered the innate immune system. So he's considered like the father of immunology, the ancient, primitive, innate immune system. Now we know there's two components of the immune system, right? There is the cell-mediated component that makes what I call air-to-air missiles. Those are called antibodies. And then there is the innate immune system, which are the hand-to-hand combat cells that are involved with phagocytosis, putting their arms around foreign invaders, squeezing them to death, and killing them with chemical warfare, which basically is the release of bleach, hypochlorite, which is what these cells do. They bleach invaders to death. And so he was the discoverer of this innate immune system process. With that, he then also, because being at the Pasteur Institute and being involved with microbiology, started to look at how the immune system of the human, having these corpuscles, these macrophages, were related to the microbiology of the gut. Because in the stool, obviously, you could find all sorts of bacteria, as we know, and we call it now the microbiome. And so he was looking at this interrelationship between what we now call the endogenous innate immune system of the gut, which we call the gastrointestinal associated lymphoid tissue, or GALT, and the relationship of bacteria in the gut that lived in kind of harmony. Well, we now know that the gut obviously is the seat of the body's immune system. More than 50% of the immune system is clustered around the intestinal tract. And a lot of people have asked the question, why would that be? Is it just like the universal designer of the body just couldn't find a better place to the immune system, so they just stuck it around the gut? And the answer is no. If you think about it, in the course of living, a human being will eat about 20 tons of foreigners, unless you're a cannibal. So we're eating foreign molecules. We just did, right? Those are molecules in our plant and animal foods that are not native to our human body. So our process of digestion and assimilation has to separate friends from foe in order to absorb friendly stuff and leave behind unfriendly stuff. So the body's immune system is there to really make that decision about how things at Guardians of the Gate get into our bodies as friends and not as foe. So the immune system is really there to defend us against foreign entry. And so about 70% of your antibodies from your cell-mediated immune system that float around in your blood are produced by your GALT and the mucosal-associated lymphoid tissue that you heard Stephen talking about earlier, the MALT. And then we also know that this immune system is very adaptive to the changing environment of the gut. So all this kind of fits together then with a book that Metchnikoff wrote later, which was called The Prolongation of Life. This is a very interesting book. It was written in French in 1902 and published in English in 1904. I actually have a first copy of this signed by Metchnikoff in my book collection. It's one of my prized part of my old book collection. And this book really is quite powerful because he talks about aging associated with the gut microbiome. It wasn't called the microbiome yet, but that's what he was talking about. And he then proposes that the treatment of aging would be to make a healthy gut by rectal installation of enemas with Lactobacillus bulgaricus, right, yogurt. So that's in this textbook. It was the early kind of concept that if you could get the gut microbiome right, it would have a favorable effect on conditions that we associate with healthy aging. So Metchnikoff was quite a philosopher and talked philosophically as a rationalist, a romantic rationalist of his age. If you read some of his writings that were published in English, they're very, very interesting, in which he really coined the term gerontology, the study of aging. Gerontology was really one of his processes. And in 2016, it marked the centenary of his death, and has been given now credit for being really the discoverer of the immune system, the innate immune system, which now has gone and been in resurgence. It was kind of lost in its importance for about 50 years, but over the last couple of decades, people now recognize that this innate immune system, this phagocytic system, is extraordinarily important for defending us from infection, but also reconstructing the body on injury. It has the ability to recycle damaged tissue. So he also was credited with speaking about something you're going to hear much more in detail after my talk, which is called autointoxication. And I'm going to say a couple of words about that just as a warm-up for the subsequent conversation. So autointoxication is this construct that somehow within the gut, all these metabolic processes are going on. You could call it fermentation or putrefaction. Those are kind of terms. And as you know, some of the chemicals that are made by that process of microbes eating debris in our intestinal tract produce molecules whose names are like putrescine and cadaverine, right? So just the names alone don't sound that wholesome, do they? And so the internal surface area of our intestinal tract is about the size, if you flatten it out, of a doubles tennis court, just to give you an idea of the size. And as Sid Baker talks about, it's filled with vomit and shit, basically. So it's a really kind of nasty environment. And so the gut is there, as a mucosal barrier with its immune system to help protect. And it's got this mucus layer. And the mucus layer is laid down by specific types of gut microbiome bacteria living in concert with the community, the ecology of our gut itself. So this is a really robust giant campout with multiple thousands of different campers with different names and different tents and sleeping bags and backpack equipment, all living in this extraordinarily diverse community. And what can happen is by imbalance, if you think about the fact that there are 3 general classes of these campers, these bacteria that live in there, these protists, the 3 types would be considered, well, I'll go from the good to the bad. The first would be considered the symbiotic bacteria. The symbiotic bacteria, let me just give you an idea. You've already heard some of these numbers previously, but I'll just reinforce them. In a gram of stool, there are about 10 to the 14th bacteria in a normal stool, which means there are more bacteria in a gram of stool than stars in the known universe. Think about that for a second. It's pretty impressive. Of that 10 to the 14th bacteria in a gram of stool, on a healthy individual, something like 10 to the 8th, 10 to the 9th are friendly bacteria, symbiotic bacteria, and commensal bacteria. The commensal are the second family of bacteria that live in the space. They don't do much good, but they don't do much harm. They just kind of hang out. So you got the symbiotes that are producing vitamins and tropic factors and immune stimulating things, and then you got the commensals. Then there's about 10 to the 4th bacteria out of the total number that are called the parasitic bacteria. And these are deconjugating. They produce secondary metabolites like cadaverine and putrescine. These can be bioactive substances that can have effects on the nervous system and on the immune system. And so we consider those to be small in number. If you think of 10 to the 14th, 10 to the 10th, or 10 to the 4th is not that many. It's a very small percentage. But these can be pretty pathogenic if they, certain forms of E. coli or Listeria monocytogenes, or there are certain bacteria that are really quite toxic. So you have to, the campers that represent the symbiotic and commensal families keep these other hostile campers in check, basically. So this whole construct of a community and ecology then gives rise to our more modern definition of the microbiome and its complexity and how it interrelates. Its personality relates to what we eat, where we live, what we think, what we're exposed to, including things like drugs that influence the orally consumed drugs that influence the microbiome. If there is an imbalance, says this 4-part series of articles that was recently published last year on autointoxication, then the imbalance can produce too many of these funny chemicals that then produces a toxic reaction that kind of overloads the ability of the body to manage them, and now you get extra intestinal effects, right? Systemic effects, because it can be absorbed into the bloodstream. Now, earlier today, we heard about hepatic encephalopathy, right? And that's an interesting thing if you think about it, because you recall hepatic, liver, encephalopathy, brain. So the liver is connected to the brain. But what's the origin, as you learned this morning, of hepatic encephalopathy? It's the intestinal bacterial debris. So it really should be gastrointestinal hepatic encephalopathy. They're all interconnected. So how would you end up with psychosis, hallucinations as a consequence of this process? You could either have too much of the bad things coming from your gut, or you could have too poor an ability of your liver to scrub it, or you could have too permeable a blood-brain barrier that allowed these things to get in, or it could be all of that you see in hospitalized older age people that are hallucinating. And what's the treatment of choice? Historically, it's been to give them lactulose. Now, what does lactulose do? It produces diarrhea. And that's a way of doing top-down flushing. Basically, it's a detox program for hepatic encephalopathy. So all these things, when you start looking at them from this lens, this perspective, kind of start to make sense, that the gut is in a constant equilibrium with our immune system and with our nervous system, and we're in this subtle balance of how we eat and how we think and the relationship to our microbiome. Okay, so that's this autointoxication concept, and that connects the gut to the brain, and you're going to hear much more about that over the course of the remaining part I'm going to discuss and subsequently. Now, autointoxication sat kind of quietly as a concept until, in Britain, the singular most well-known physician, the physician to the king, Sir William Arbuthnot Lane, took this concept up very seriously, this autointoxication concept. He was considered a surgeon extraordinaire. Some of you probably have read either about him or know his biography. Really advanced surgery and developed all these techniques that are even still used today in surgery. And he took up in the concept of autointoxication as being a concept that had clinical validity. And so that, because of his position of authority, people like William Osler, who then traveled from England to the United States to be a principal physician setting up the first British-like medical school in America, which is Johns Hopkins University in Baltimore, brought this concept to the States. And so autointoxication kind of transferred over the Atlantic, and it became, in the early 1900s in the United States, a concept that particularly related to psychiatry because of the brain-gut connection. We all okay so far? So everything's looking like there's this lineage of thinking, and people are buying into this model. Well, one person that really bought into this model, full hook, line, and sinker in the United States, is Henry Cotton. Now, Henry Cotton was a psychiatrist of extraordinarily big importance in the United States because he turned out to be the head of the Trenton State Mental Institution in New Jersey in the States, which was the largest mental hospital in all of the United States in the early 1900s. It had over 10,000 patients, so this would be a huge facility. And Henry Cotton was a strong believer, going through this lineage I've just talked about of autointoxication. The problem, well, before I tell you the problem, I'll tell you the issue. The issue is that because of his prominence and because Trenton was close to the seat of our government in Washington, D.C., and so it was very visible, he got lots of government support. He became very well known in notoriety in medicine, and his concepts then influenced all these other outlying hospitals and medical centers around the United States to believe in this autointoxication concept. Now, the treatment of choice for individuals with this condition of this time was surgical resection of the bowel. So they started taking out portions of the anatomy, right, to treat this. So psychiatry became a surgical procedure. And then he was giving these presentations to our government about the extraordinary success of these treatments, that people that had been schizophrenic and all these very serious mental institutionalized patients were getting well. And he was publishing this work and giving presentations at national meetings. And so all these other facilities, that wanted to be like the New Jersey medical facility were adopting the same procedure. Well, it turns out that about 20 years went on, or maybe not quite 20, about 17 years went on, and then finally there was a whistleblower that had worked with Henry Cotton that said that he had found huge amounts of doctored information. His statistics had been made up. There's erasures on patients, and many, many people that didn't just fail but actually died from the post-surgical complications were not even reported. So when they went in and they did a big congressional hearing, and this became a national scandal, it turned out that not only was it not useful, but it was like they had a huge number of deaths from, because they were taking out everything. They started with the tonsils and they just went on down, and if the patient didn't respond, then they just went to the next surgery. Some people had 20 surgeries. And so what happened is that, when people kind of get the reality of what was going on, they didn't just vilify Henry Cotton. They vilified the concept of autointoxication. They said, oh man, this is like witchery. This is horrible. And so in American medical training, from the year of about 1940 until the 1980s, those 40 years, if you even mentioned autointoxication in a traditional medical institution, you'd either have to have a prefrontal lobotomy or be purged to a foreign island somewhere in the South Pacific. it was not accepted at all. And this whole story I've just described, by the way, is beautifully told by a medical historian at the University of California, San Diego Medical School, Andrew Scull, in this book called Madhouse that he wrote a few years ago. That's like a medical mystery because it traces this whole history really elegantly and how this was covered up and all these statistics were doctored and so forth. The point I'm trying to make here is that one never really looked at the concept of autointoxication in terms of its physiology. It was really related to the mischievous actions of Henry Cotton and the death of all these people that then threw it out entirely. So it basically stayed quiet for many years. Well, then for me personally, and this is where it all kind of started in the early 80s, where I started seminaring on this concept. I was unaware of this Henry Cotton work until much later, but I happened to meet Dr. Bjornsson from Karolinska in Stockholm at a meeting when I was over here in Europe in the early 80s. And Dr. Bjornsson is a gastroenterologist, internationally regarded, who was the first person to develop this term leaky gut. And he was measuring leaky gut in two types of individuals, alcoholics and in people taking NSAIDs. Now we all know that non-steroidal anti-inflammatories have a real problem with gut permeability. Back then it was not known. And he published these early papers in The Lancet on these gut permeability issues. And he even suggested that the reason that there are so many problems with liver disease in alcoholics is because the alcohol increases the permeability to toxic substances that then damages the liver. So this was in the early 80s. This is one of those papers, Leaky Gut of Alcoholism. So when I saw this work and I spoke with him, I thought, wow, this is very interesting because I was aware of Metchnikov's work, and I thought maybe we're seeing a new way of explaining what Metchnikov was talking about. So we started early in the 80s talking about this, and that led actually to the development of the first laboratories to start measuring microbes in the stool, basically, stool testing in the early 80s. So that led to the resurrection in the United States of this concept. And I take just a small little part of this story, a pride of this story, because there were many others in the United States who became principals to be kind of first-line resurrectors of this concept. The first name in that list is a remarkable gentleman. He's passed away now, Arthur Caswell, a gastroenterologist in Santa Barbara, California. And when I first started to think about this, I had just happened, you know what they say, chance favors a prepared mind. I went to a meeting in which Caswell was speaking, a medical meeting, and he was talking about the fact that he was doing stool testing in his patients because he learned so much from the stools, like undigested protein and fats and microbes and so forth. And everybody should be doing stool testing if they really want to understand the health of their patients. That was his advocacy. And I thought, wow, that's kind of interesting. Well, at the time, I was, this would have been the late 70s, I was the director of a medical laboratory that serviced the largest kind of preventive medical clinic in the Pacific Northwest called the Bellevue Renman Medical Lab. So I came back and said to my lab group, we had to start doing the stool testing. One of the people in my study group at that time, in the evenings we met once a month, was Jonathan Wright. Jonathan Wright, some of you know his name, Dr. Wright, very prominent physician, had his own laboratory associated with his clinic, so we then started to do stool testing. One of those students at the first naturopathic college in Seattle, which was Bastyr University, was founded in 1978. One of those first year students was a gentleman by the name of Stephen Berry. Stephen Berry then did an internship while he was a third-year naturopathic student with Jonathan Wright and with my lab, and he ultimately then took his practice to Asheville, North Carolina to become Great Smokies, now Genova, which then became the large stool testing laboratory. So this all kind of happened over a number of years, you know, that this started rolling forward with technology. And of course, Marty Lee was the lab director for Great Smokies, a very well-known clinical biochemist. And one of the major consultants was Leo Gallen. Many of you know him, an internist from New York City, was very prominent in understanding these relationships. So there are lots of people that were kind of bringing this concept back into clinical practice in our field. Still, by the way, I'd have to say, when I spoke even in the 90s about this at medical meetings, it was like considered some kind of witchery or quackery or something. You know, it was not was not usual and customary. One of the other individuals I want to give credit to, who was very important in this, was Joe Pizzorno, who is the founder of Bastyr University, and really brought the science of natural medicine more prominently into the development in America of naturopathic medicine. And he was a big proponent of this as well. And then lastly, Patrick Hanaway, who became later the medical director for Great Smokies and Genova, and advanced this farther. So a lot of interesting people in this area. Now, the reason I bring this up is that when I was at the Pauling Institute on sabbatical in 1981 and 2, I had a number of conversations, this is kind of early on in my education, about this gut microbiome connection to the immune system with Dr. Pauling. And you know, he was not an immunologist as such, but some of you probably don't know this, but he is credited as being the father of immunology from the discovery of antibodies with Delbruck when he was at Caltech. And so if you go back to the early history of the development of immunology and with the cell-mediated immunity, Pauling had a very deep interest in antibody-antigen reactions and the structure of antibodies in the immune system. So when we later at the Institute for Functional Medicine thought about this, we thought, you know, we ought to really be giving an award in the name of Dr. Pauling, which became then the Linus Pauling Functional Medicine Award that we've given every year then subsequent to honor his contribution to this field. And when I look back at the years of award winners, it gives a topography of the evolution of this field. So I'm going to just quickly do if you'll bear with me, I'm going to share with you the winners of this award just to show you how I think our field has evolved. So the award is given to the clinician or researcher who has made a significant contribution to the development of functional medicine. And so here is a kind of a litany. The first award winner was Glenn Doman for the achievement of human potential in Philadelphia with brain-injured children and their families, and really finding how to manage brain injury based upon a complex network effect of gut, diet, and exercise. And second was Leo Galland, you probably know, very important. Third was Gerald Reven with insulin resistance and metabolic syndrome. Next was David Perlmutter with functional neurology. Next was Abram Hoffer with orthomolecular psychiatry. Next was Walter Willett from Harvard with the whole nature of diet as a treatment for chronic disease. Next is Joe Pizzorno. Obviously, I've spoken with him. Next was Catherine Wilner from Mayo with chronic with neurology. Next was Eleanor Rogren from Nebraska with women's hormones, metabolism, and gut function. Next, Michael Hollick with vitamin D. You probably know his work. Next was Bethany Hayes with the woman's hormonal system and its relationship to microbiome. Next was Mark Hyman, some of you know is now the chairman. Next was Dean Ornish, who has got a huge contribution to lifestyle medicine. Then they stuck me in there. They had to find a filler. And then next was Bruce Aim in orthomolecular nutrition, and Mimi Guarneri with cardiovascular function. Next was Alacio Fasano at Harvard, with the gluten connection to brain function. Next is Randy Jurtle with nutritional epigenomics. Next is Bob Roundtree. Many of you know of his work as a premier faculty. And Toby Cosgrove, the founder and chairman of Cleveland Clinic. And lastly, Patrick Hanaway. And then in 2018 was Terry Walls with her work on neurological function. So that, if you really take those people and look at them over the last 25 years, that kind of outlines how this field has evolved, starting through the pioneers and the developers of the field. So that takes me down to leaky gut as a danger signal in autoimmune disease. Because once you have a permeable gut, then foreigners, larger molecular weight substances, can transverse across the gut, and now they have entry into or exposure to the immune system. So if they're unfriendly, the immune system is going to respond as it should. It's there to defend us. And depending upon the genetic uniqueness of that person, they may have more vigorous response or less vigorous response. So there's a systemic effect of permeable gut that activates the immune system. And it can be seen in sites of former injury. Like you're an athlete and you got a leaky gut, then that knee you injured 20 years ago suddenly starts getting swollen. And you say, well, why is my knee bothering me when it hasn't bothered me until I got this gut infection when I was traveling in India? And so we're starting to recognize that the immune system has a memory. And if you have the breakdown of these barriers, I call it the firewall. It's the gut firewall. So if the firewall breaks down, now you have an ability to be exposed to things that your immune system recognizes as foreigners, and it does the job as it should, but sometimes it may overdo a good thing. And now you get collateral bystander activity, which means tissue injury to your own tissues. Okay, so leaky gut, leaky brain. So if you got a leaky gut, does that mean other membranes are leaky as well? And the answer is yes, they're correlated. We actually in the United States learned that during the Gulf War, where servicemen and women were exposed to all sorts of different toxic substances, and we found that their guts were permeable, a combination of stress of being in warfare plus toxic things they exposed to in the Kuwait deserts. And they also ended up with a lot of neurological symptomatologies. And we had a number of those come into our clinic. We saw about 4,000 patients a year on different research projects in our clinic in Washington State, because we were very close to Fort Lewis and McChord military bases. We got a lot of veterans coming in from the Gulf War, and many of these individuals had very severe neurological sequelae that they were sure were results of some exposures during the war. These are men and women that were very, very fit, high performance individuals that came back very disabled after the exposure. And we were able to show, using lactulose mannitol testing, their guts were permeable, and they had neurological oxidative injuries, inflammation, neuroinflammation. So leakiness is not just the gut itself, it may be other membranes that serve as barriers of defense as well. And the microbiome plays some role to these systemic metabolic effects. And why do I say that? Because we've done quite a bit of work over the years. I put up one paper there that we published with Patrice Connie and his group at Catholic University Louvain, in which we showed that there are certain phytochemicals that would actually help to resist permeable gut or leaky gut. And so if you had a diet that was really deprived of phytochemicals, meaning a plant-free diet, and it was a lot of inflammatory fatty acids, and as you probably know in the saturated fatty acid family, what is the long-chain saturated fatty acid that has the greatest inflammogenic potential? It's palmitic acid. It's C16-0. And palmitic acid wasn't very high in the diet actually until we started feedlot feeding beef. And when you feed corn at beef, you get them, because they have, remember, they're ungulates, they got all these bacteria in multiple stomachs, and they make a lot of palmitic acid, and that gets absorbed into the meat. And so we start getting high palmitic acid meats, which then enhances the inflammasome. It enhances inflammation. So now it increases gut permeability and increases the inflammatory potential. So I don't think we could say meat in and of itself, beef in itself, is inflammation producing, but the way we've raised meat maybe is a contributor. And we found that phytochemicals, in this case we were studying what are called the isohumulones. Isohumulones are found in a variety of plant foods, one of which is hops. And so we did a lot of work with hops, these lipophilic compounds found in hops, and we found that it could resist then the breakdown of gut mucosal integrity. And so those of you that are hoppy beer drinkers, you knew there was a reason for it, didn't you? So I'm now giving you permission to be an IPA drinker. But there are many other phytochemicals that have positive effects, and I'll talk about them, particularly the polyphenol families that help to maintain gut mucosal integrity and are actually food for certain microbes in the microbiome that then enhances mucosal protection. So when you eat a high plant-based diet with a variety of different colored plants, to use Dr. Minnick's concept, the rainbow diet, then you're getting a lot of these nutrients that the friendly bugs are going to convert into gut salvaging secondary metabolites. Okay, so of microbes and meals, the health consequences of dietary endotoxemia. You're going to hear much more about endotoxemia, but just suffice it to say, this is where the gut mucosal barrier breaks down. You start absorbing molecules that activate the immune system in a way that can produce systemic effects that are far away from the gut itself. They can be at the brain, or they can be in the heart, or the lungs, or in a variety of tissues. So you see a variety of different dysfunctions associated clinically with endotoxemia. And that's the breakdown of these GI tight junctions, the barriers that hold the gut mucosa together. And you probably recall that Alessio Fasano was one of the first people, if not the first, to discover that there is a peptide, a protein that binds these together called zonulin. And zonulin is kind of a glue substance that helps to maintain the tight junctions. And so when you start changing your zonulin concentrations, which actually can be measured clinically in the stool, it can be associated with alterations in gut mucosal integrity. Okay, so there are many mechanisms that have been now well explored as to why this concept of gut dysbiosis. Now I want to take a moment here just to stop. So when I first started talking, and my colleagues started talking about dysbiosis and postprandial endotoxemia, I can tell you this wasn't a friendly reception we were getting to the broad community. We were not getting invited to any parties. We were off the guest list. This was really considered heretical to any kind of good thinking by the standard gastroenterology community of the time. And now I'm talking about even into the middle 90s, probably even into the late 90s. This concept was that I'm describing to you now, which is now being seen in the standard literature. If you do a PubMed search in the Library of Medicine, over the last 10 years, you'll see literally hundreds of papers in top-tier journals on dysbiosis and gut permeability. But these were not even terms that would be allowed in social conversation before. And I think the reason for it, again, was a legacy of this period of what happened with the Trent endotoxemic autointoxication thing. But fortunately, new mechanisms were discovered and new ways of actually describing this, and it became more socially acceptable, and then grants were given by National Institutes of Health that made it fashionable to do this research, and people were pulled into it. And once you get fashionability, then everybody wants to be part of the community. And so we started to see this thing grow up. And of course, Fasano and others were very principally important in its developing its understanding at a mechanistic level. Now, when we look at the literature, and this is just a paper that appeared in Science magazine this last year, just to show you that these are getting in the top-tier journals, gut microbiota at the intersection of diet and human health. They talk about, again, these diagrams you see all the time, the breakdown of the gut mucosa, the absorption of these molecules across the lumen, and they get through the hepatoportal circulation. And what's the first organ that is intersecting the hepatoportal blood as it comes from the gut? It's the liver. And what do we now know in our world of today, in our developed world? Now that we've got these new medications that treat successfully Hep B and Hep C, now the major cause of liver damage and ultimate need for a liver transplant and liver cancer is what? It's called NASH, non-alcoholic steatohepatitis, and non-alcoholic fatty liver disease, and NAFD. And where do they come from? They come from stuff that comes from the gut that affects the liver in such a way that it produces fibrosis and ultimate injury because of oxidative stress. So this is Helmut C's work, hepatic oxidative stress caused by toxic reactions that the liver is trying to detoxify, but it's injuring itself in the process, right? I'm kind of using broad brush, probably not very precisely, but to give you a general pattern of what's going on. So we now know this is socially acceptable to talk about this, and that leaky gut ties together with diet quality, and it is also related to what we consider the metabolic inflammatory archetype of obesity. Now, there's been a long-standing debate, and I'm not going to try to resolve the debate. I have a certain feeling about it, but I'll just tell you the debate. The debate is around, does obesity cause inflammation, or does inflammation cause obesity? There are literally thousands of papers that you read in the literature that say obesity causes diabetes, obesity causes heart disease, obesity causes cancer, obesity causes hypertension, it goes down the list. And I started asking a question, and this is now well over 20 years ago, saying, are we sure that we know that it's a causal effect? It's associated for sure, no question. But could it be that obesity is a part of the sequelae of something else that's upstream of that actually has obesity and this condition as covariables? Maybe it's something else upstream that happens to demonstrate in the phenotype both obesity and meaning metabolic interruption, and what we call metabolic inflammation, or inflammaging. Some of you have heard that term, inflammaging. So I don't want to get into the debate because we could push and take on this quite a bit, but my theme, I think, if I could give you the bottom line of how I think about it, I think it's a combination of both. I think obesity can create an inflammatory potential if you have aggravated, I call it angry fat. You can have friendly fat like in Santa Claus, and you can have angry fat that produces a different immunological state of your endocrine system. And people with a high body mass index, the biggest part of their endocrine system are their fat mass adipocytes, right? That's the biggest part of their hormonal system. And so obesity can contribute to that, but also conditions that relate to inflammation can contribute to making fat angry. So it's a push-pull, cyclical dog chasing its tail type of argument, I think. Okay, so with that then, we then say, well, what about this microbiome? This is a very complex community of all these organisms that are camping out and trying to vie for space and get their nutrients worked out. So there's now more and more evidence to say that these microbiome are fingerprints just as unique as our genes. The difference is that the genes of our microbiome are probably 10 to 100-fold more diverse than the genes of our eukaryotic cells, because we have all these species of microbiological organisms in our gut and elsewhere in our body, all of which their own genetics that differs from our genetics, right? So you have way more DNA that are not eukaryotic DNA, that are protist DNA, than we have DNA of our own body. And they're all signaling different messages and cross-talking. And some people say that our body's genes are just really being regulated by our microbiome. It's really the gut function that controls how our mammalian genes, so we're really a product of our microbiome. I think that's a little bit probably exaggerated for my position. I think there's a communication that goes on that is in dynamic tension between our microbiome and our host genes, but certainly they're in direct relationship. There's no question about it. So the gut microbiome is our second genome. It dynamically integrates signals from the host and its environment that impacts our health and disease risk. That's absolutely for sure. And personalized modulation of the microbiome by nutrition using various types of pre- and probiotic interventions are now being demonstrated to be powerful therapeutic ways of normalizing microbiomal function that influences gene expression of the host and influences our phenotype. Did that make sense when I said it all like that? So it all kind of fits together as a network. So people ask you, I'm sure, as you get, particularly at this stage of my career where I've been doing this a lot of years, what are you most proud of? Well, I'm most proud that I've been around for 40 years. That's probably the most proud thing. But of those 40 years, there's been a few things that I really am really proud to have been associated with. And right at the head of that list is when we developed the concept of the 4R program. And I remember that day so clearly. It was in our clinical center in Gig Harbor, Washington. The Institute for Functional Medicine had just been formed. So this would have been about 1993, I think. So it was 1991 that we formed the IFM. And we were having a conversation about gut restoration and this whole theme that I've been sharing with you. And I said, the way that we've been doing this in our clinic as an experiment, to trying to find a therapeutic intervention, involves these various 4 steps. And then when I said the 4 steps, I recognized that they all had an R as the first letter. And I said, hey, this is the 4R program. And it's the remove, replace, reinoculate, repair program. And everyone said, yeah, that sounds good. We can all remember that, the 4R program. Now I think we went to the 5R and the 6R. Now there's lots of R's out there. But it started off as the 4R program. And people often ask me, if I had one thing to learn as a skill in the art of this, where would I start? And this is where I would start, because this program will reproducibly improve people's health so they will come back as your champion very rapidly. If properly deployed and executed, this in a matter of a month can really turn a person's phenotype, their health around. So this is a great place to start. There are 2 principal therapeutics that I think are good skills to gain. One is the 4-hour program, gastrointestinal restoration, and the other is hepatic detoxification. Those two things constitute a baseline way of approaching function of people with chronic illness across all sorts of different diagnostic codes. So detoxification, hepatic detoxification, and gut restoration, kind of principal therapeutic tools. So let me ask you, how many people here have not heard of the 4-hour program? Let me see. Oh, fantastic. That's great. So you're all skilled in the art, or at least you're familiar with it. And this is something you can learn to be good at very quickly. And a lot of people say, I don't know where to start, and I feel apprehensive because I don't know enough. Let me put it this way. I've been doing this 40 years. I don't know enough. I still feel that apprehension. I feel still like, oh, there's so much more to know. But I know enough to do the 4-hour program, and it'll work and make me look like I'm really good. So, you know, that maybe is enough just to start, to gain your self-confidence and to get good patient feedback. Now I'm going to jump off here for a second, and I would guess that no one in this audience has ever heard this next 15 minutes. So this is new, and you have to bear with me here, but I think you'll find this interesting. You may think it's a little esoteric, but I hopefully will show you why I think it's worth your thinking about. So we have this view in genetics that there are these monogenetic mutational injury diseases that we call genetic metabolic diseases of infancy. Tay-Sachs, Wilson's, Gaucher's, Fabry's, sickle cell anemia. You can go down the list, right? These are these monogenetic diseases that you can track with dominant and recessive Mendelian genetics. And what we believe, or what we're trained to believe, I think, is that these diseases are immutable because they're hardwired into our genes. And if you do a genetic test and it comes up with one of these as the Monte Carlo bad luck of the draw, you say, oh my word, that's really too bad. There's nothing we can do about it because you've got this genetic disease. You ever heard this before? Okay. And I think to some extent, that's true. But I want to kind of indulge your thoughts for a moment here to consider that maybe these things are not as immutable and fixed and deterministic as we thought. That even these monogenetic diseases have some variable in how it's expressed in the phenotype. Even things like Down syndrome. You can have highly functional Downs that are happy children that grow up and have a pretty good life, actually, maybe even better life because they don't seem so stressed. And then there are other phenotypic presentations of Downs that are very serious and the children don't do well. So the same gene, this trisomy 21, can display itself in many different phenotypes. And I would believe that, I know that that's true for phenylketonuria. That's true for sickle cell anemia. You could go down the list of any one of these monogenetic metabolic disorders, and you can find a wide variation in how children are affected. Now, with that in mind, I want to take one to show you how this connects into a broader thinking, and that's sickle cell anemia. So sickle cell anemia, you know, is this very severe problem where the red cells become packed with a funny form of hemoglobin that doesn't work with its neighbors very well in the red cell. The red cell is about 75% of the volume is hemoglobin. And so these hemoglobin molecules are a slightly different shape than non-sickled hemoglobin. And so they don't fit together. What they do is they crystallize into a kind of a lattice work that then distorts the shape of the red cell so it goes from its normal discoid shape into what looks like a sickle. And then the sickle cuts its way through the vasculature. So it produces all these inflammatory processes across the body. It doesn't just affect one tissue. It can produce in some people heart disease, other people neuromuscular, it affects virtually every organ in different ways. So we would say, well, that's too bad. If you got the dominant, you got your two recessive genes together, and there's nothing you can do about that. You're going to have these sickling events, and they are episodic crises, and that's the way it is. Well, years ago, it was found there was a drug that could treat sickle Cell Anemia. The problem is the drug had a fairly bad toxicity side effect, but the drug is called Hydroxyurea. And Hydroxyurea, it wasn't known at first how it worked. It was just kind of observed that patients who took this drug seemed to have less sickle crisis than if they didn't take the drug. And over time, more and more studies were done on the mechanism of action of hydroxyurea, leading to this New England Journal of Medicine paper last year, or no, it's this year actually, early this year, talking about the mechanism. And here is where the story becomes very interesting and why I thought you'd find it intriguing. It turns out that infants who carry the sickle cell gene do not have sickle crisis when they're first born. It only presents itself until they grow a little older in infancy and into toddlerism. And the reason for it is when they're born, the majority of their hemoglobin is fetal hemoglobin. Fetal hemoglobin is not sickled. Okay? What's sickled is adult hemoglobin. And it takes a while for that to grow in after the child is born. Even, however, as an adult, we still have some fetal hemoglobin in our blood. It doesn't completely go away in terms of its expression. It may be less than 10% of our total hemoglobin, but there's some fetal hemoglobin. The amount of fetal hemoglobin you have in your blood will dilute the sickled hemoglobin, and if it gets up to something like 20%, it really lowers the risk of sickle crisis because it kind of buffers the blood against sickling, or at least the sickle crisis. So the concept was, how could you increase fetal hemoglobin expression, because those genes are still in your genome, they just went silent. That would be an interesting biological treatment for sickle cell anemia. And it turns out the drug hydroxyurea actually increases fetal hemoglobin. Okay, so that sounds pretty cool. But the problem with the drug is it has not a very good safety window, so it has a lot of other effects that are not so good. So let's follow this through for a second. In examining this, now that we have all this genomic capability where we can measure genes much more successfully than before, it turns out that there are certain genes that regulate fetal hemoglobin production. One of those genes is a gene that's been called BCL11A. That gene suppresses fetal hemoglobin's expression. So here is a case history of 4 people that were given hydroxyurea who all carry sickle crisis, and the red circles I put around is what impact did it have on this when they took hydroxyurea, what impact did it have on their BCL11A gene expression. And you'll notice in all cases it's low relative to the other bars that are higher. And what are the other bars? Those are the expression levels of fetal hemoglobin. So by suppressing the expression of that gene, it opens up the potential of the body to make more fetal hemoglobin. And these 4 people, all with the sickle cell crisis, then had lower sickle cell episodes. So I'm tying molecular genetics to a drug to a phenotypic outcome. Are you getting where I'm going here? Okay, so the reason I'm going into this is that this gene is going to have some interest here in a second. Hold on with me. So then they said, well, but there are people that have hydroxyurea-resistant forms of sickle cell crisis, but they will respond to the administration of sodium butyrate. Sodium butyrate. Now, when I saw that in the literature, I immediately thought to myself, oh my word, what's the major source of butyrate in the body? The gut microbiome, right? Specific species of bacteria that make short-chain fatty acids, butyrate, propionate, and acetate. So then I got wondering, could it possibly be that there's some connection between gut health and sickle crisis in people with genetic carriers that would modify the expression of this phenotype into something? They would still carry the gene, but it would not be expressed because their sodium butyrate produced endogenously could hold at rest their expression of the sickling hemoglobin. That's what this paper is all about. It starts talking about sodium butyrate. So can you then treat sickle cell anemia with butyrate? Ta-da! The answer is yes. So the gray bars on this particular graph, these are all patients. These are 11 patients that all have sickle cell genes. The level of butyrate that they have before the administration, the percent of fetal hemoglobin they have before administration of butyrate are the hatched bars, the gray ones. Then on top of those is the amount of fetal hemoglobin produced in them after they're administered sodium butyrate. Can you get the drift of what I'm saying here? So you notice there's big increase in fetal hemoglobin after these people were given, all 11 of these people were given butyrate. Now this has been given to them exogenously by infusion. Okay, now with that in mind, how does that relate to the vaso-occlusive crises that we see in sickle cell anemia? This is the biggest risk that sickle cell carriers have, is the risk to their vasculature and heart disease. So what do you think they, like, just to read the title of the paper, it really says it all, right? Sickle cell vaso-occlusive crisis, it's a gut feeling. It's a gut feeling. Meaning people that have dysbiosis and carry the sickle cell gene have many more crises of much greater severity. And it's tied through the linkage with these genes that suppress fetal hemoglobin that's associated with butyrate produced endogenously by the gut. Now that begs the question, doesn't it? Well, gee, could we take a person then with sickle cell gene and give them something that improved their gut restoration and increase butyrate endogenously and reduce sickle crisis? Could that be a therapy? Good question. And the answer is yes, we can do that. This is all literature that's been published in which reduction in gut immune activation in sickle cell animal models was achieved by giving them a gut restoration with probiotics. So we were able to demonstrate in a sickle cell animal model that this seems to work. Then we go on into human studies and we say, what about probiotic supplementation and sickle cell associated vaso-occlusive disease? And if you look at this little diagram up here, what do you see? You see intestinal microbes, short-chain fatty acids, butyrate endogenously, going through the gut mucosal and increasing the immune system activity, suppressing gene expressions of the repressor of fetal hemoglobin, increase in fetal hemoglobin production, dilution of the sickle hemoglobin, and reduction in crisis. The gut health has something to do with the modulation of a genetic metabolism disease through gene expression effects. Okay, that's enough. That's enough. You hung with me. I appreciate it. That was a lot of left hemisphere of the brain stuff. But the reason I went through this, I want you to understand when we say network medicine, how these things are interconnected. They're interconnected in ways that you only understand if you dig more deeply. Now, the question that I would ask, and you know this is kind of somewhat of a rhetorical question, how many medical students, even in gastroenterology, get associated with this little 15-minute lecture? I would say it's approaching 0. Now, did I have to go and make up these studies? No, these are in the readily available peer-reviewed literature just by going to the library. There's nothing like magic about this. What's magic, if there is magic, is you have to ask the right questions. The questions you ask determine the answers you get. We're in a syllogism of a do loop in medicine right now because it's all pathophysiologically focused, assuming that the genes are deterministic and you can't do anything about them, so don't even ask the question. Never ask the question, you never get the answer. That's a duh. I'm telling you, you were just exposed to something that 99.99% of the intelligent, well-educated, gastrointestinally specialized community wouldn't have a clue what I just said. That's the fact. Now, am I criticizing them for that? No, what I'm saying is it's hard to know what you don't know. It's hard to know what you don't know. And when you tell a person who has undergone an extraordinary number of years of service and training and specialization and testing and expense for education that what they don't know is what they need to know, the first response is, who the hell do you think you are? Right? Because who wants to have to go back and read all this literature? You already know the answers. Stop making life confusing. So, someone once said to me, I'm sorry, I'm going to be a little bit probably facetious here, but not totally, so bear with me. So a question I have commonly asked of me is, well, Jeff, you give up these great stories and, they're very entertaining, but I want to see the randomized clinical controlled intervention trials because I practice scientific medicine and I would not do anything that had not been RCTized. So if you can show me the RCTs, then I will be persuaded that maybe what you're saying is more than just an interesting story. So my response now, after many years of this debate, is to say the following. So let me assume, just for the sake of our discussion, that I'm not practicing by your convention a completely scientific medicine. So if I acknowledge that, let me ask you a question, if you'll give me the same honesty. Okay, what's the question? The question is, do you ever have a patient in which you prescribe simultaneously more than one drug at a time? Well, yes, of course I do. And do you have that patient on that drug for some extended period of time? Oh, yes, I often do. I might use an antihypertensive, anti-lipidemic, an anti-anxiety. I might use a variety of different medications. Good. Then you're a purveyor of scientific medicine. Absolutely. RCTs. So can you give me the RCT that defines those drugs in that patient for that duration and gives me the outcome? And if they're honest, they'll say, well, no, I have a study for this drug and I have a study for that drug. I said, no, that's not what I'm asking. I'm asking those combination of drugs in that patient over that period of time that you're going to administer it. Well, no, of course I don't have that study. I said, well, good, then let's both admit we're practicing non-scientific medicine, except mine is safer. Yeah, so I'm sorry, that's a little bit of a flip, but that's, the kind of aggravation I get after these 40 years. I'm less an apologist than I used to be. Okay, so immune cells in the gut may explain why some people can't lose weight. This is, the work I was talking about with Patrice Connie and Nathalie Delzin at Professor Riboffau's laboratory at Louvain that has really revolutionized now. Many other labs around the world are studying different microbes and their influence on calorie control. We at first thought it was that the gut bacteria were eating up calories. And that's a very small part of the story. It's really related to the effect that these microbes have on the enteroendocrine system. Now, what do I mean by the enteroendocrine system? I'm amazed that we still haven't really talked about this as much as I think it deserves in this field. So, as you know, along the length of the 7 meters of your gastrointestinal tract, in different regions of the gastrointestinal tract, on the surface of the mucosa, you have different receptors. They're not the same receptors on the whole length of the... So you have ghrelin receptors like proximally upstream, and then you've got GLP-1 receptors at the L cells of the distal ileum. And so you have different receptors in different places that pick up different information. The L cells of the distal ileum, these cells are specifically picking up messages from bitter substances in your diet. And they are receptors that are called TAS2 receptors. There's 25 different TAS2 receptors in mammals, of which some of those are directly related to control of the release in the L cells of enteroendocrine hormones, such as GLP-1. And what does GLP-1 do? Once stimulated and released into systemic circulation from your gastrointestinal mucosal cell, it then circulates in the body to the beta cells of the endocrine pancreas to regulate insulin secretion. At the peripheral site of the insulin receptor substrate 1 on the cells of your myocytes and myocardium, it activates insulin sensitivity, and it serves as a downregulator of the cassette of genes that control inflammation. So here is a hormone that's produced by your GI mucosal cells that gets responded to by certain triggering substances in your diet that happen to have certain bitter taste attributes, which we've cut out of the diet because kids don't like bitter, and therefore they only like sweet. So only ghrelin gets activated, and ghrelin activates appetite, and GLP-1 reduces appetite. So the whole physiology of the endocrine nature of food and eating is connected into our food supply and connected into the regulation of these hormones that then do things like I'm going to now give you new information just published from our group last February. It turns out that the ovaries and the testes, so women ovaries, men testes, have on their surface bitter taste receptors. So the ovaries are tasting. Now you're going to say, come on. I mean, you know, why in the world would the universal maker of the body throw bitter taste receptors on the ovaries? That's ridiculous. Well, it's not ridiculous because these are not bitter receptors, they're chemosensory receptors. We just label them as bitter. They pick up all sorts of other information, not just bitter substances like DAMPs and PAMPs, like pathogen-associated molecular debris. So they are signaling receptors that tell organs what's going on outside. So if a woman has dysbiosis, what does that do to her fertility? It lowers her fertility, right? Because her ovaries learn that her body's under hostile attack and it's not a great environment to have a child. We have you better save, this is the salvage technique, right? Save critical functions. And so these chemosensory receptors, which we call bitter receptors, are really there to signal the environment and then to establish gene expression in the individual tissue as to how it's going to function. And it turns out, we have discovered and published, that the bitter receptors on the ovaries trigger the gene expression control of cytochrome P450C17, which is the rate-controlling enzyme for steroidogenesis of androgens in the ovary that regulates things like PCOS, that regulates polycystic ovary syndrome. So what I'm saying is we're living in this extraordinary, interesting network of biology in which we have separated this biology out into individual chapters of a textbook in which we study the reproductive system by itself, close the book, take a test. Then we move to the gastrointestinal tract, study it, close the book, take a test. Then we study the immune system, close the book, take a test. We don't see that all these things are interconnected and talking with one another. And you can only get to the source if you understand the communication, not who was communicated with. Am I making sense to you here? So, and then that brings in the most important components of how diet and lifestyle are interacting with those receptors, which, by the way, may start in the gut with your microbiome signaling to your gut mucosal receptors. It's ecology. It's a giant home that we're trying to make as a friendly place by turning on the genes of bliss and turning off the genes of alarm. It's a very simple thing said, but very difficult sometimes to tease apart clinically. Okay, so that leads us into fecal microbial transplant. Now, when you heard this the first time, what did you think? I mean, really? When I heard for this the first time, I thought, oh man, this is going to go down like a lead balloon. I mean, so they won't even accept dysbiosis or postprandial endotoxemia. Now we're talking about fecal transplants? Oh, my word. So look at where this journal is, where this publication. It's in America, considered one of the premier journals, the Journal of the American Medical Association, January of this year. And what's the title of the paper? Effective fecal microbial transplant on 8-week remission in patients with ulcerative colitis. An RCT, a randomized clinical control trial. So this is a great time to be around. It's a great time to be around because a lot of the things that we said for which we were vilified, castigated, or thought to be a subhumanoid are now suddenly becoming like the topics of great discussion. In cocktail parties, you talk about fecal transplants. It's amazing. My kids will tell you, I don't want to hear any more about feces at the dinner table. But now I'm saying, think of how advanced you were in the 3rd grade when we were talking about this. So, you know, it's a very, very interesting time. So what is the impact of the microbiome and diet on inflammatory diseases? I think you already can jump ahead and know the answer to this story is that now all these papers are being published that show these same diagrams of permeable guts and stuff leaking through and activating the immune system. And if you look up here, you'll see liver, muscle, and adipose tissue. These are signaling tissues that receive that information from the gut. So each one of those has a different metabolic effect on their function. So the adipocyte, which we used to think was this kind of sloppy cell that just engorged itself with extra calories called lipids and just stuck around for a rainy day that never came, now we see is actively involved in the production of what we call adipokines. And these adipokines are inflammatory mediators that speak to tissues at a distance. And what do we know are their homologs from their embryogenesis? The microglia. And where are the microglia found? We used to call them the glue that holds the neurons together in the brain. But actually, they're the brain's immune system. And they're communicating with the macrophages that are communicating with the embedded white cells in the gut, which are in the liver, which are called what? 10% of the liver are immune cells called Kupffer cells, right? So the gut mucosal cells, the immune cells in the gut, speak to the immune cells in the liver. They're the same brethren. Those are called the Kupffer cells that speak to the circulating macrophages that speak to the embedded immune system in the brain, the microglia. And that's why gluten reactions cause dementia. They're all connected if you understand the mechanism. It's not just by happenstance. Are you getting this? Okay. So, microbiome metabolites, many different types of metabolites that all influence function at a distance. I'm not going to go through those in great length, other than to say that we now know that substance that naturopaths were measuring years ago, and I recall in the 70s, actually, I had this discussion about the way you could qualitatively measure endotoxemia using a test called the Indican test. The Indican test is you take a urine sample, you treat it with hydrochloric acid and this dye, and if the urine turns blue, that means you have this substance in the urine, which is a tryptophan metabolite associated with endotoxemia called indoxal. And for years, this was just kind of a naturopathic lore. It was a qualitative thing you could do in your office. In fact, the company was formed based on my seminars, was called Eurotox. It was actually selling this kit to doctors for many years. Now, most recently, this is 2018, they're finding that this uremic toxin indoxal sulfate is associated with gut dysbiosis and bacterial putrefaction, and it is a qualitative test for damage to the kidney and damage to the vascular system. So it also relates, obviously, as we've already talked about, to the liver, because the liver is the first organ of insult, like in LFT and NASH. So is this a debate? Not really a debate, but now the question is, what kind of diet is best? And the answer is yes. That's my simple answer to a complicated question. That there is no one diet that's perfect for all people to restore their microbiome. And some people need different therapeutic diets than others. That's why we have a whole array of therapeutic options that we personalize. And obviously, I don't have the time right now to go into this great detail, but this is part of what you all become masters of that most people know nothing about in medicine. They have not studied this, so they don't know this. Now, one of the people that has studied this extensively is a colleague of mine, Stan Hazen, who's at the Cleveland Clinic, and he's top of his game. And he's the guy who has gotten a lot of international notoriety for his discovery of TMAO, trimethylamine oxide, which is a byproduct of endotoxic or let's call it microbiome metabolism of choline. And this choline metabolite by gut microbiome called TMAO is a vascular toxin. And so there's a big debate. Are meat diets bad? Because choline is much higher in animal products than it is in vegetable products. So there's a big debate about, if you have high TMAO, should you go on a vegan diet? It's high in fish, so people will say, oh, you don't want to eat a lot of fish. There's big debates, but what we do know is that TMAO production is very individualized to the person's own microbiome. So some people can eat a lot of meat and fish and have very low TMAO. Other people can eat a vegetarian diet and have high TMAO. So it really relates more to your microbiome than it does specifically to your diet, but there are interrelationships of how they cross-talk and communicate with one another. So what we're starting to see, and you'll notice the organs that are listed here in this diagram, are the multiple organs that the immune system is influenced from what comes from the gut into systemic circulation and influences their function. Now, there's another good friend I have, Elaine Chow at UCLA. This is her work that has been studying seizure disorders and the ketogenic diet. And what do you think she has found, published recently and gotten a lot of international notoriety for this work, that the ketogenic diet works in seizure disorders because it influences the gut microbiome to alter signaling substances that come across the gut that have brain effect on the low frequency activity of neurons that produces seizures. She can actually make animal models of seizure disorders, like epilepsy, happen by altering their gut microbiome, and then she can treat that by putting the animal on a ketogenic diet and show that the ketogenic diet modulates the microbiome, which then affects the neurological sensitivity. So we're learning more and more about how these things fit together, and that leads us into fasting mimicking diets, right? This is the Walter Longo work, extraordinary work showing that you can regenerate the immune system through hematopoietic stem cell rejuvenation by putting a person on fasting mimicking diet programs. And you can clear a bunch of junk that was accumulating in the immune system that relates to this metabolic inflammation. So now primary therapy in medicine is starting to become nutritional intervention, medical nutrition therapies. We're crossing the boundary into a new era. How much of this is taught in medical schools today? Still very little. That's why you're the vanguards, you're the leaders, you're the people that are taking this out clinically. FMD, fasting mimicking diet cycles, partially reverse IBD-related pathology. So again, having positive impact on the immune system of the gut. Fasting mimicking diets promote the expansion of the friendly bacteria, like Bacteroides and Lactobacilli, that are associated with friendly commensal and symbiotic bacterial activity in your gut. So, man, oh man, this is like the new age. All sorts of tools that can be employed now for modulation of these chronic conditions associated with dysbiosis. And then that relates to this group that I started to work with closely recently, the Eran Siegel group in Israel. That's this extraordinary discovery that they're making and publishing almost on a monthly basis, this work, to personalize the diet based upon the glycemic response that they can predict from the gut microbiome. From a stool sample, this group has developed an algorithm in which they can predict what your glycemic response will be to various foods in your diet without ever doing continuous glucose monitoring. And it actually syncs up like 90% if you were to do continuous glucose monitoring and how your body, you individually, will respond to those foods, whether they're cooked foods or raw foods, whether they're canned or frozen. They have all, I think it's something like 24,000 foods that they've studied now and catalog these in this algorithm that has huge predictive ability. And they're now really trying to get this to be used routinely in diabetic treatment centers to actually help a person design their personalized diet that would improve their, it's not just glycemic index alone. So this is not just like glycemic index of food. This is personalized based on the way your microbiome responds, how it influences your blood sugar managing system. This is a powerful new age, right, in which we're harnessing these tools. And in fact, in this recent paper that was just published in JAMA, they took a group of individuals, this is work done at Mayo actually, they took a group of individuals that did not have diabetes, and they asked the question, could they use this gut microbiome analysis, the Eran Siegel group's analysis, to predict how non-diabetic people would respond to their foods in terms of their glycemic response? They then put continuous glucose monitors on them to measure. They did a prediction from their gut microbiome, and it came out, as I said, 90% predictability based on the personality of their microbiome. Yeah. Yes, this is a new era, isn't it? So I just want to close. You've been very patient and tolerant with me. I've waxed and waned over a whole topography here of as my family would say, Dad, you can make the most simple things so complex that no one understands them. I have a very great skill in that area. But we have what I think is coming up, a remarkable meeting in Seattle this October 11th and 12th. I know that's a long distance for you to come. We will be posting later the videos of this presentation, but you want to come to a meeting that's really talking about where the tire meets the road and this new technology. That's our meeting coming up in Seattle, October 11th and 12th, in which we're really going to be drilling deep into this personalized nutrition. What's the frontier? How do we use these types of devices? This is an Oura Ring, but many other devices. How do we use this information? How do we use big data? How do we actually start to go from description to actual prescription of diets with a great degree of specificity? That's our objective in our October meeting. And I think we're going to assemble some of the great minds to really work on this in this process. And as you know, the doubling time of information in this field right now is less than 3 years. Meaning if you graduated at the top of the class 3 years ago and you just took a rest for 3 years, you'd be intellectually stillborn today. So this is a really hugely dynamic time to be a part of this field, and you're our leaders, and keep it up. Thank you so much.